Let's talk a little bit about the neuroeffector junction. So the neuroeffector junction, we talked about it loosely before, is the synapse between the second neuron, the postganglionic neuron, and the actual effector organ or effector tissue. And so it's really going to look different than the neuron to neuron synapse. So basically the preganglionic neuron and the postganglionic neuron, that synapse, it's also going to look different than skeletal muscle synapse. So a neuron, we only got one neuron in the somatic nervous system, which is a whole nother story we'll talk about. But basically the synapse here looks different from the somatic nervous system and from the neuron to neuron synapse in the autonomic nervous system. This is called the neuroeffector junction. Some of the differences here is that we actually have these swellings along the end of the neuron. So we don't have the discrete axon terminal that we've kind of been drawing, the terminal that we see at a neuron to neuron synapse. We actually have just these varicosities that are these swellings that have neurotransmitter in them that are found along the end of the postganglionic neuron. And so when the action potential gets to the end of this postganglionic neuron, it's actually going to trigger the release of this neurotransmitter from these varicosities. And the events here are pretty similar to what we've been looking at, but the setup is slightly different. And we'll talk about some of those differences here in a bit. All right, so we're looking at the neuroeffector junction. Which is only in the autonomic nervous system. And so let's kind of just illustrate this out here. Briefly, we've got a preganglionic neuron. And this is that discrete axon terminal that I've been talking about, okay? So at our initial synapse, remember this is the autonomic ganglia here. I'll just put AG. And at the end of this postganglionic neuron, we don't have this discrete axon terminal. What we see is that we've got the endings of this axon, and then we've got these swellings along the endings here, along the nerve endings that are called varicosities. Okay, this is only found along the postganglionic neuron. So this is the postganglionic. Okay, and these are varicosities. Actually, let's draw that somewhere else. Let's put in here the tissue that's being okay. So here we actually have our effector cell. Let's draw smooth muscle here. This can also be cardiac muscle or glands. Let's actually list that out here. So cardiac muscle, just so we're clear what the options here are. Glands or adipose, okay? And so this is the neuroeffector junction, this region here, okay? And it looks very different from this synapse. It also looks different from the synapse between the motor neuron and skeletal muscle, okay? So let's kind of zoom in on the varicosity and the smooth muscle and the synaptic transmission between those two structures. So we're zooming in here on the neuroeffector junction. We've got our large swelling, which is the varicosity. And we've got our effector cell here. Okay. And so we know that the action potential propagating all the way down this axon, it's going to get to the end of the axon, and it's actually going to trigger up the same type of events that we've seen at a traditional synapse. So it's going to influence voltage-gated calcium channels on the membrane. such that calcium rushes into this varicosity, and we've got these vesicles stored in the varicosity. Now, this is where things can get complicated. So remember, this can be a parasympathetic neuron. This can also be a sympathetic neuron. So there are two options for this neurotransmitter here, depending on where we are in the autonomic nervous system, okay? So I'll put it up here that this can be acetylcholine if we're in the parasympathetic system. Remember, both parasympathetic neurons release acetylcholine, or it can be norepinephrine, epinephrine if we're in the sympathetic system. Okay, so there are two options for what this neurotransmitter in the vesicles here can be. Either case, the calcium channels open up when that action potential gets here, calcium rushes in, and we've got that exocytosis that we talked about. So exocytosis, such that we dump this neurotransmitter, whatever it is, into the cleft, okay? Or the junction here. Now, on the other side, we've got a receptor that's going to bind this neurotransmitter. Again, there can be some options here based upon what we've talked about. So this can be an adrenergic receptor if the neurotransmitter is norepinephrine or epinephrine. And this is where we have all the options we talked about: alpha 1s, alpha 2s, beta 1, beta 2, beta 3. Lots of options, lots of different tissues that we can actually be talking about here. And then obviously, remember, the second messenger is going to differ, right? This only uses IP3, sorry. And these three are going to use cyclic AMP. So just making that connection of the actual mechanism here as well. The other option for this receptor is that it can be a muscarinic receptor, which is also a cholinergic receptor. So again, make sure that you're keeping those details straight, right? Which is a cholinergic receptor here. And that is also going to generate a response. So we have a couple different options depending on what the neurotransmitter is, which really depends on what division of the nervous system this neuron is found on. So when that neurotransmitter binds onto this receptor, that's how we get our second messenger generated and our cellular response. So all of this leads to cellular response. Okay. And then we also talked about the acetylcholinesterase. If this is a junction that has acetylcholine, so if it's a junction that has acetylcholine, then we're going to have that acetylcholinesterase enzyme here. Right? Remember, acetylcholinesterase, we talked about it before, it's found at this junction as well, because this junction always has acetylcholine. All preganglionic neurons always release acetylcholine. So on the surface of this neuron, we're always going to find acetylcholinesterase. But here we have some different options, right? If this is a junction that has norepinephrine and an adrenergic receptor, we're not going to find acetylcholinesterase. In other words, acetylcholinesterase cannot degrade norepinephrine, so we will not find it at a junction that has an adrenergic response. All right, so I'm going to put here muscarinic junction, right? Actually, let's put cholinergic, because this could also be at the neuron-to-neuron synapse. Right? So only at cholinergic junctions. Okay. And we talked about the rest of the mechanisms for clearing the junction. So we can have the neurotransmitter diffuse away. It can be broken down and then reuptaken. So let's remember our reuptake molecule here on the other side. That can help to recycle neurotransmitter here. But this can also just diffuse away, right? We talked about some of those options earlier. In the case of norepinephrine, it will always diffuse away because there's no enzymes such as acetylcholinesterase, there's no synonymous enzyme that breaks down norepinephrine or epinephrine, and so it always has to just diffuse away from the junction in order to clear the junction. Okay. So that was quite a lot. This is what happens between our varicosity, which is at the nerve effector junction only, and our effector cell, which is smooth muscle, cardiac muscle, glands, or adipose. And these are some of the options for the neurotransmitter at this junction, and for the receptor and the second messenger response at this junction as well. Okay. Again, I'll pause here just briefly and see what questions you have, and then we'll keep going. Let me add here as well, while you kind of think about questions, let's add here some details as well. So acetylcholinesterase actually breaks down acetylcholine into acetate and choline. Oops. All right, so it breaks down acetylcholine to acetate plus choline. And that's kind of how it kind of degrades it further and kind of assists in the clearing of the junction, okay? All right, so let's kind of keep going here. Okay, so here is a really nice representation or illustration of the two options that we have for this neuroeffector junction. So I talked about the fact that it could be at a postganglionic parasympathetic neuron, right? So that's going to be releasing acetylcholine and binding to a muscarinic receptor. Or it can be a postganglionic sympathetic neuron where it's going to release norepinephrine and that's going to bind to an adrenergic receptor, of which there are many options or subclasses here. Okay, we've also got acetylcholinesterase only at a cholinergic junction, which breaks down this excess acetylcholine to choline and acetate, and that can kind of provide some further options for clearing this away from the junction, which is so important for ceasing the signal of this, the transmission of this signal. Now on this image, you'll see the mitochondria with monoamine oxidase or MAO. Don't be worried too much about that, but this is just where norepinephrine is made. So that's kind of the connection here of putting that in this image is showing where norepinephrine is made. And then we don't have that on the acetylcholine or the cholinergic junction. But other than those differences, the signal transmission is pretty much the same. We still have calcium entering the varicosity. We still have the diffusion or exocytosis, and then the binding of the receptor. The general principles are pretty much the same. Okay,